looks like we have good number of people who are joining, and the numbers are constantly increasing. So let's go ahead and start. Welcome everybody to this week's Connect Science seminar. I'm very excited to introduce our speaker today, Dr. Stark, Dr. George Stark, a distinguished scientist at the Cleveland Clinic Foundation. Dr. Stark really needs no introduction. He has made several significant contributions to science, and one of the most notable and definitely one of the most exciting contributions that he has made is that of developing two of the most important techniques that any molecular biologist would be aware of, which is the northern and western blots techniques. And he has made several important contributions to the discovery of the JAK-STAT pathway. And also his current research direction revolves around understanding the complex pathways on how human cells respond to interferons and to the activators of STATs and NF-kappa B. And that is, I think, what we will be hearing about today, including his recent work on PDL1. Just a couple of housekeeping things. Please type your questions not into the chat option, but into the Q&A option, and we can have Dr. Stark answer them towards the end of the presentation. Without too much delay, I'd like to give the floor off to Dr. Stark. Please take it away.
Thank you, Balaji, for that nice introduction, and also for helping me to participate in this very exciting new series. I think it's a fantastic idea. So, as the title says, I'm going to tell you about recent work in our lab and others, but especially focusing in this talk on the stuff that we've done, how we've come to appreciate very interesting and complex new participation of interferon in cancer. And to start, I need to show you a bit for the first few slides about the basics of interferon biology, especially in the context of infectious disease, because that lays the foundation for what I want to tell you later on. So as you all know, I'm sure, interferon drives are major antiviral pathways. In context of infection, it's synthesized in response to viral dsRNA or RNA. A very important point, which many people do not appreciate, is that the way interferon works is that it primes uninfected cells to die rapidly upon infection. It really does not protect cells against virus infection, quite the contrary. It induces cells to die when the virus invades them. So in that way, it prevents the virus from replicating and proliferating. And for these reasons, the prime state is not sustainable, and so the response is downregulated very quickly in about a day. But we've learned that a special mechanism, which I'll tell you about, preserves a residue of the antiviral activities that are not toxic for a much longer time, so the cells can sustain that. And this mechanism turns out to be highly relevant for the role of interferon in cancer. So just a brief summary of interferon signaling. I'm going to be talking, incidentally, only about type 1 interferon and not about interferon gamma or type 2 interferon. So the JAK-STAT pathway was discovered in the context of interferon signaling, and interferon binds to a 2-protein receptor, activates 2 tyrosine kinases, TYK2 and JAK1, leading to the phosphorylation of the STAT transcription factors, STAT1 and STAT2 on specific tyrosine residues, and then together with IRF9, that complex in the nucleus drives the expression of 100 or so genes that encode the proteins that constitute the major antiviral response. This is a crucial observation from our lab that really led to a lot of what I'm going to tell you about. For whatever, I can't remember the reason anymore, but for whatever reason, we decided to look in this Western at interferon signaling over a long period of time. So we're looking not at the first couple of hours, but out to 2-3 days. And what's boxed in red is very important. So as I mentioned, the initial phosphorylation of the stats here, shown as stat 1, is very quickly downregulated and is basically disappeared at these longer times. But notice the very substantial increase in the level of expression of the transcription factors, stat 1, stat 2, and IRF9. This experiment was done in 2 non-tumor human cell lines. But we've seen it in every cell that we've looked at. So we wondered, what in the world, why is this happening? Why would the, in this time scale, would the concentrations of these transcription factors increase, but without tyrosine phosphorylation anymore? What might they be doing? Well, one thing is that they're still conferring antiviral activity to the cells. So this is an experiment in which we're using HME cells, which I say overexpress, but really at similar levels to those induced by interferon, either the wild type or even the Y to F mutant proteins that can't be tyrosine phosphorylated, and no interferon treatment. This is just expressing stat 1, stat 2, and IRF9. And you can see that alone confers a 2 to 3 log increase in the antiviral activity in response to 2 RNA viruses. So this is nothing like the full response to interferon in protection, but it is something, it is substantial, and that's probably why this mechanism exists in nature. So very quick summary of this: interferon is synthesized, it phosphorylates the stats. Many of these proteins are toxic, they're quickly downregulated, but a small subset of these interferon-induced proteins is sustained for many days, and that continues to provide partial protection against infection. And the expression of these genes is driven by the forms of stat 1 and stat 2 that are not tyrosine phosphorylated, plus IRF9. So now to interferon in cancer after that brief introduction. Interferon is present in the tumor microenvironment. It turns out it is produced both by immune cells and, very importantly, by the tumor cells themselves. And it has 2 disparate effects: high levels of interferon, which can be produced in cancer cells in response to DNA damage, or administered therapeutically, as was done in the early days of interferon, inhibit the tumor. But as I'll show you, low and sustained levels of interferon are crucial for tumorigenesis and benefit the tumor. So the big difference in how the cells respond depends upon the level of expression, and that is regulated. So as I mentioned, high doses of interferon were initially used to treat cancer. Ralph Weichselbaum's lab, who's done a lot of interesting work in this area, showed in this paper that if the interferon receptor is knocked out, the cells are sensitized to ionizing radiation. In other words, the production of interferon by cancer cells in response to DNA damage caused by the radiation helps to kill them. And if you prevent the cells from responding to that interferon, they do better. I'm sorry, they do worse, right? So the cells have to, I'm sorry, I messed that up a little bit. The cells have to respond to the interferon that they produce in response to DNA damage to help them to help them survive, sorry. And so the conclusion is that a high level of interferon administered therapeutically or produced in response to DNA damage actually helps to kill the cancer cells, right? So the Weichselbaum lab also found what they called the interferon-related DNA damage resistance signature, which I'll abbreviate IRDS, and the references are down here. And this is a subset of about 1/4 of the interferon-induced genes, so 25 to 30 genes, that have been upregulated in many types of cancer that are resistant to radiation and chemotherapy. And the expression of these genes in many cancers correlates with increased tumor growth and metastasis, and that leads to a poor prognosis. So this information was published without any knowledge of what the mechanism might be, but we quickly found that these IRDS genes were this very same genes that were increased by the higher level expression of unphosphorylated STAT1, STAT2, and IRF9. And the green just shows the coincidence in different cell lines between the genes identified by the Weichselbaum lab in cancers and the genes that we found whose expression was sustained over a long period of time by unphosphorylated STATs. So it's the same, this is the phenomenon that's responsible for the IRDS phenotype. And to model what's happening in cancer, we took normal fibroblasts and treated them with a very low level of interferon over a longer period of time. And although after 2 hours, there is a response, you can see the phosphorylation of STAT2 here over longer time. When we treat them with this low level for a long time, the phosphorylation of the STATs goes away, but you can see the big increase again in STAT1, STAT2, and IRF9. So this experiment tells us that sustained exposure to a low level of interferon sustains a steady state in which these 3 transcription factors and the downstream proteins maintain high expression, essentially for many, many days. And this just shows the representatives of the IRDS subset of the interferon-induced genes whose expression is high after 16 days. And these are the non-IRDS genes, the majority of the genes whose expression is not sustained by the unphosphorylated STATs. So this IRDS phenotype is seen in many cancers. It's driven by the high expression of STAT1, STAT2, and IRF9, which in turn is driven by constitutive exposure of cancer cells to low levels of interferon. And this interferon, as I've mentioned already, can come from the microenvironment or from the cancer cells themselves, which is what we're going to be talking about next. So about a year ago, Hanju, Annalise, and I published this paper in PNAS showing a novel role for PDL1 in helping to sustain this chronic intrinsic response to interferon, which incidentally enhances resistance to DNA damage, as I hope to show you. So let me remind you very quickly about PDL1. I'm sure you all know about this. It's an immune checkpoint protein. It's expressed on many cancer cells. It suppresses T cell activity by binding to PD-1 on T cells. And the expression of PDL1 is regulated in a variety of ways, some of which are mentioned here. But it turns out that some important functions of PDL1 are independent of T cells. So in cell culture free of the immune system, PDL1 as a cell surface protein mediates resistance to radiation, chemotherapy, and as you'll see, interferon. And here are some previous references prior to our own work, which I'll tell you about now, in the literature. So we're talking about the cell intrinsic function of PDL1 in addition to rather independently of its ability to interact with CD8 T cells. So to summarize this work at the beginning, so you have a sense of where it's going, we've discovered that PDL1 partially inhibits, but only partially, the response to interferon. It activates the ability of cells to make interferon, and so it functions as a rheostat or a regulator that simultaneously dampens the acute response to interferon, which cancer cells don't like because it's cytotoxic, but it helps to sustain the chronic response, which helps the tumor cells. So this is using a small cell lung carcinoma cell, which is resistant to radiation and chemotherapy and expresses high levels of PDL1 and the IRDS proteins in an in vitro system, knockout of PDL1 here. actually causes an increase in the number of dead cells, which is what we're scoring here, in response to increasing, in response to a time course, rather, with cisplatin. So PDL1, in other words, which is here and not here, protects the cells against cisplatin, and it also protects the cells against ionizing radiation, as shown in this dose response experiment on the right. So PDL1 does this by inhibiting the acute response to interferon. So here is an experiment in which we've looked at a time course, a treatment with a good-sized dose of interferon in PDL1 knockout cells. And you can see that when PDL1 is knocked out, there's a much stronger phosphorylation of STAT1, STAT2 in these cells. But notice that the phosphorylation is not completely gone. So it inhibits but doesn't eliminate the response to interferon, and that's very important. Greatly downregulated, but not absent. So how is interferon made in cancer cells? I've mentioned this briefly already, and we're particularly focused here on the role of double-stranded DNA as an interferon inducer. dsDNA in the cytoplasm, where it doesn't belong, free in the cytoplasm, activates an interferon synthesis pathway through the receptor protein, which is called cGAS, which uses ATP and GTP to make a cyclic dinucleotide with 2' and 3' linkages, which is a signaling molecule, which activates the effector protein, which is called STING. So STING leads to the phosphorylation of IRF3, which activates it, and also the release of NF-kappa B from I-kappa B inhibitors. And these two transcription factors together then coordinate the synthesis of type 1 interferons in the nucleus. This is only one of two major mechanisms by which DNA synthesis by which interferon synthesis is stimulated, the other responding to double-stranded RNA, which we're not so concerned with here. So Balaji, can you, I'm seeing on my screen, a participant has enabled closed captioning. Can you get rid of that? I can't affect it. Oh, yes, I can. Sorry. That was me, not you. Okay. So
okay, great.
Yeah, I'm trying to get, because I can't quite see all the titles. Now it's better. Okay, so interferon produced by cancer cells is induced through the cGAS STING pathway. Here's an experiment in which we've knocked down cGAS, and you can see the resultant loss of phosphorylation of STING, although the expression of STING is not affected, and loss of phosphorylation of IRF3, and loss of the synthesis of interferon. This is gene expression at the RNA level. And this pathway increases the expression of both PDL1 and IRDS in a subset of cancer cells. So here's PDL1 expression. You can see is very sensitive to the presence, to the response to cytoplasmic DNA in the cancer cells. And the resultant loss of expression of the IRDS proteins, as you'd expect. This is at the protein level with OAS1 and OAS2, and at the RNA level with these three proteins. All of these are IRDS proteins whose expression is sustained by the unphosphorylated STATs. And this is an experiment done in small cell lung cancer cells. We see the same phenomenon in a different cancer cell line. So PDL1 provides two things. It provides positive feedback that can sustain cell intrinsic interferon beta expression, leading to IRDS expression. So here again, if you knock down the interferon, I'm sorry, if in the PDL1 knockout, there's loss of expression of interferon, and in this loss of expression also of these 3 IRDS genes, including PDL1 itself. I'm sorry, this is a PDL1 knockout. It's a different experiment. So when PDL1 is knocked down, then the phosphorylation of STAT1 is decreased, as I showed you, and so is the expression of the IRDS proteins. So this is one of my favorite experiments from recent years, and what it shows is that the presence of the interferon beta gene in cancer cells is very important for cell survival. So in the control cells, we're staining the dead cells in green in this incucyte experiment. In the control cells, you can see there are very few green cells, of course. But if you knock down interferon, you can see that most of the cells in this experiment are not able to survive when the interferon beta gene is knocked down. And the control experiment is over here. So if you take these knockdown cells and expose them exogenously to interferon, if you give them the interferon outside that they can no longer make themselves, the survival is much better. And this is plotted quantitatively here. These are the controls. This is the interferon knockdown cells, looking at percentage of dead cells now over as a function of time. And then at 2 different concentrations of interferon, you can see that there's very substantial protection of cells. And we've seen this in a couple of different cell lines now. So at least for these cells, we can call these cells addicted to interferon. In other words, in order to survive, they need the protective effect of the low levels of interferon that are made in response to DNA that the cancer cells are producing themselves, probably in response to errors in DNA synthesis and other aberrant mechanisms. So the summary of this part of the talk, which is up here, is that PDL1 can block the, but not entirely eliminate, the response to a high level of interferon, protecting the cancer cells from the cytotoxic effects of a lot of interferon. But in cancer cells where the cGAS sting pathway is constitutively activated and cell intrinsic interferon beta expression is going on, these chronic interferon responses increase the level of PDL1 and increase the level of the IRDS proteins. So that makes more PDL1, which can sustain the inhibition up here, but can also help the IRDS expression, which sustains cell survival. So this is basically a positive feedback loop that promotes both the cancer cell intrinsic sting activation, leading to both interferon beta and IRDS expression. And as I've already mentioned, high levels of PD-L1 suppress the cytotoxic acute interferon responses. So PD-L1 enhances resistance to radiation and chemotherapy independently of the immune system, is what we believe. And we call it the Goldilocks effect because, you know, the cancer cells want their interferon response to be not too cold, and not too hot, but just right. So that's the job of PD-L1. So this next part of the story is the discovery made by Sarmishta and Elise, and again published in recent PNAS about a year ago, that the E3 ligase, FBXO22, is responsible for degrading PD-L1 and for sensitizing cancer cells to DNA damage and to anti-PD-1. So standard therapy for non-small cell lung cancer, which is the most predominant cancer in the world includes immune checkpoint blockade, but as you can see, only about 30% of the patients respond at all to this therapy. So there's an effect here, but there's certainly a lot of room for improvement. And we focused on this disease in our preliminary studies. Now, the discovery of the E3 ligase was made using a novel genetic method that we've pioneered a long time ago in collaboration with Mark Jackson and Andrei Gudkov. And I'm just going to mention this briefly. This method is available in the literature for review, and we've used it effectively in a number of different contexts. But basically, we use insertional mutagenesis to put a promoter using a lentiviral vector more or less randomly into a large library of cells. And then we select those cells with some sort of a stress. In this case, we've used ionizing radiation to kill the great majority of the cells, but IR-resistant mutants survive. And then we can now take the total pool of RNA from this pool of resistant mutants and put that pool through high throughput sequencing to identify upregulated genomic RNAs that are linked to the vector RNA, because the mutagenic vector causes the synthesis of a, if you like, bicistronic or complex RNA that includes vector sequences. So by simple sequencing and focusing just on those sequences in the pool, that genomic sequences that include vector sequences, we can generate a list of strong candidates that might be responsible for the phenotype, and then chase those down in detail later on. So when we did that experiment using ionizing radiation and the insertional mutagenesis, what we found was a clone related to FBX022, which is an E3 ligase. And what was cloned in the first experiment was just this piece, the C-terminal piece, truncated fragment of FBX022, which turns out functioned as a dominant negative in the genetic experiment. And when we cloned the full-length FBX022 and put that into cells, we found the opposite phenotype. So full-length FBX022, when overexpressed, increases the sensitivity to ionizing radiation. And then because, as you saw earlier, we saw a connection between PDL1 expression and sensitivity to DNA damage, we looked next to see whether there was any effect of high expression of FBX or knockout of FBX on PDL1. So in this experiment, in A549 cells, when you overexpress FBX, you see a dramatic loss of PDL1 expression. And when you knock it out, you don't see very much, but there is something, because the expression is already pretty high. Similarly, it's not as dramatic, but I think quite substantial in the FBX overexpression in CALU cells, another non-small cell lung cancer cell line. When you overexpress FBX, PDL1 expression decreases very substantially. We think that FBX directly or indirectly, probably directly, mediates the ubiquitination and degradation of PDL1. So in this experiment, in these two cell lines, we've used a proteasome inhibitor, and then IP'd PDL1 and then blotted with an antibody to ubiquitin. And you can see in both of these cells, there's a tremendous increase in the level of ubiquitination of PDL1 rather in the cells that overexpress FBX. And coming back to the origin of this experiment, if we downregulate PDL1, we sensitize the cells to DNA damaging agents, either ionizing radiation or cisplatin. So this is a dose response for both of these treatments, comparing, in this case, the PDL1 knockouts, or knockdowns, I should say, with control cells. So how is FBX able to decrease PDL1? What's upstream of FBX that might be regulating this pathway? And you might ask the question, if you're familiar with what goes on in the minds of oncologists when they confront patients and evaluate the PDL1 levels in their tumors, they find that low PDL1s in a tumor predict that the immunotherapy using either anti-PD1 or anti-PDL1 is not likely to help. So why do we want to knock down PDL1 when tumors without PDL1 are not responsive to immunotherapy? It seems counterintuitive, right? But we think that it's likely that these tumors still evade the attentions of CD8 T cells, but by a different mechanism. So PDL1 is not there. The immune cells have to find another way to resist CD8 T cells. So it's no use to try to manipulate either PD1 or PDL1. So immunotherapy is justifiably restricted to a subset of patients in which there's substantial or significant expression of PDL1. In other words, the target is there. So we can still consider strategies to downregulate PDL1, but only when the levels are significant, because if they're not significant, chances are there's some other mechanism involved for resistance to T cell killing. Okay, and we got a tremendous hint from this paper published a few years ago in Science by colleagues at Case Western Reserve, showing that CDK5 is very important to attenuate PDL1 expression and promote anti-tumor immunity. So what is CDK5? Despite its name, CDK5 is not a proline-directed serine threonine kinase. It is a proline-directed serine threonine kinase. It's not a cytokine-directed kinase. That was an error in the original nomenclature. Nevertheless, high expression and aberrant activation of CDK5 occurs in many cancer types, including non-small cell lung cancer and facilitating tumorigenesis, and high expression of CDK5 correlates with poor prognosis and shorter survival of non-small cell lung cancer patients. So here we're showing that knockdown or inhibition of CDK5 increases the levels of FBX and decreases the level of PDL1. So this is knockdown in again, these same two non-small cell lung cancer cell lines. You can see that when you knock down CDK5, you greatly reduce the level of PDL1, and you greatly reduce the level of FBX. So if we can inhibit CDK5, we can expect the same phenomenon. So here we've used a drug, which is, I'll show it in a minute or two, in clinical trials, roscovatine, which inhibits CDK5. And you can see that inhibiting the activity of CDK5 increases the level of FBX and decreases the level of PDL1. So here's the pathway that we can then imagine that a CDK5 inhibitor will increase, will by blocking the activity of CDK5, or over here by knocking down CDK5 in a different experiment, proof of concept, will increase the level of the E3 ligase and therefore decrease the level of PDL1. And so if you do that, if you can use roscovatine or here looking at the knockdown of CDK5 or the use of roscovatine to show that when you do that, you also affect sensitivity to DNA damage. Here, showing the sensitivity to cisplatin. So inhibiting CDK5 with roscovatine increases the sensitivity to cisplatin greatly. So the summary of this part is the E3 ligase FBXO22 destabilizes the PDL1 protein. CDK5 in turn destabilizes FBX. So if you inhibit CDK5, you will increase FBX and decrease PDL1 expression, and that leads to sensitizing the cells to DNA damage. But what about the response to anti-PD1, to immunotherapy? So can we use CDK5 inhibitors in combination with either anti-PD1 or anti-PDL1 to downregulate PDL1 and therefore sensitize cancer cells to CD8 T cell mediated lysis? So if cancer cells have high PDL1 and they're using that mechanism to resist being killed by CD8 T cells, can we get rid of the PDL1 and sensitize them? Can we use roscovatine as a selective CDK5 inhibitor? Well, roscovatine has been in the clinic for quite a while. It's a cyclocell drug. It's been in clinical trials, even in non-small cell lung cancer, in phase 2 trials. It hasn't been wonderfully successful, but it hasn't really been combined with immunotherapy, because the initial premise of using this inhibitor, which is also an inhibitor of some of the CDKs that regulate the cell cycle, is that is the effective mechanism of action. We think that the important mechanism of action is the inhibition of CDK5, which is not a cyclin-dependent kinase, despite its name, as I've mentioned. Okay, so there's certainly the ability to go ahead with roscovatine in a clinical trial, should it prove effective. So we've done a preclinical model experiment, as shown here, starting out with Lewis lung carcinoma cells. This is a syngeneic mouse model, which have been luciferase labeled, and a small number of cells is injected percutaneously, directed into the lungs of the mice. We monitored the tumors by luminescence, and then the tumor-bearing mice were then randomized into 4 groups with a small number, only 5 mice in each group in this initial experiment. And they got either vehicle or roscovatine or mouse anti-PD1 alone or mouse anti-PD1 plus roscovatine. And using luciferase, we monitored the tumor volumes. So here's the result. And you can see that this is now looking at the imaging. You can see that in all cases, I'll show you the survival in a minute, but you can see that in all cases, in surviving animals, the tumors increased until the death of the animals. In all 3 cases, except for the case when the mice were given both roscovatine and anti-PD1. And what was even more remarkable in this, so it's a tremendous effect, long-term survival of the mice, but even resistance to a new inoculation of the same cells. So now the cells in which the tumor uniformly takes are completely resistant to seeing the tumor again. So these mice have acquired resistance following this treatment with anti-PD1 and roscovatine to reintroduction of the same tumor by the same method. And this is the survival data. Again, all of the control mice and the ones that received either roscovatine or anti-PD1 alone survived, but a significant fraction, 2 2 out of the 5 mice rather died, 3 survived, and these mice, as I mentioned, completely survived rechallenge with the same tumor. So high PD-1 expression decreases cancer cell death that's induced by DNA damage as well as by T cells. Downregulation of PD-L1 using CDK5 inhibitors decrease the damage-induced cancer cell death and decrease CDK5 inhibitors increase the sensitivity of I can't see the whole slide. This is why I'm stumbling sometimes. Increase the sensitivity of the cells to DNA damage and to CD8 T cells. So we think that the use of the CDK5 inhibitors is logical for increasing the efficacy of the current anti-PD-1 and anti-PDL1 in treating non-small cell lung cancer. And so CDK5, just to reiterate, CDK5 inhibition decreases PDL1 through FBX022, which increases the sensitivity to immune therapy and increases cancer cell killing. So we would like to, of course, begin a clinical trial using roscovatine in combination with anti-PD-1 and also sensibly in combination with carboplatin, which is currently used in standard of care therapy of this disease, because roscovatine should affect both of these pathways. It should sensitize, as I mentioned, should sensitize the cells to DNA damage and also sensitize them to anti-PD-1 based therapy. We would love to do this. We're having, I might mention, quite a bit of trouble inducing the company to allow us to use this drug in a trial, which is a shame because although we can try to work with other CDK5 inhibitors, this is the one that's by far furthest along in the clinic, and we'll have to sacrifice a few years in order to work this through with a different inhibitor, which isn't so far along. So we're hopeful that we can continue to persuade the company to provide the drug for a clinical trial. But it's an issue right now, to be frank. Also, on the lab side, there are important mechanistic details that we really don't know much about. We have hypotheses that CDK5 may directly phosphorylate FBX and thus sensitize it to an E3 ligase that catalyzes its degradation, but we don't know this. So there's a big question mark here, and a lot of questions about how and where FBX can ubiquitinate PDL1. So important mechanistic details to be sorted out. And we also don't know, not shown in the slide, how in the world high expression of PDL1 inhibits the ability of cells to respond to interferon. For example, does it interact directly with the interferon receptor? That's one possibility. There are many others. So lots and lots to do on the mechanistic side. But I think we know enough to think seriously about going forward more rapidly on the therapeutic side. So an overall summary of everything that I've tried to tell you today. Low levels of interferon help tumor cells to survive. PDL1 is a master regulator that prevents the toxicity of high levels of interferon and sustains the production of low levels of interferon, this Goldilocks effect that I've mentioned. If PDL1 expression is lost, the cells are sensitized to DNA damage and to anti-PD-1. CDK5 increases inhibition of CDK5, it should say, increases FBX, which in turn decreases PDL1. That should read inhibition of CDK5. And therefore, inhibition of CDK5 is an attractive anti-tumor therapy. A lot of the information that I've told you about is now published in a review that I and my colleagues have recently put into Trends in Cancer just this month. And if you want to look at a lot more of the information and relevant references from many other labs that have contributed to this story, you can refer to this review. And Balaji, I will stop here, and I think there's plenty of time for questions and discussion.
Thank you so much, Dr. Stark. That was a great presentation. We do have a few questions in the Q&A, and then I think one spilled over to the chat. So let me just go and read them out to you. If you have the Q&A option, you can also open it as I read them from there. So first question, does the PDL1 inhibitory effect require binding to PD1?
No. So the inhibitory effect on response to interferon is carried out in the complete absence of PD1, and we've checked actually in the cancer cells themselves, they do not express PD1. So there's no PD1 anywhere in the experiment. There's no CD8 T cells, and there's no expression on the cancer cells that we've used.
Mm-hmm. So next question from Bill Forrester here. As have the SH-PDL1 studies been performed using blocking antibodies against PDL1?
Yeah, so blocking antibodies against PDL1, my colleague Hanju, who is now in Detroit running her own lab, is working with this. But first of all, the blocking antibodies, depending on which antibody you're using, have different effects. And they're not inhibitory antibodies. They simply get in the way of the interaction between PDL1 and PD1. So their effect on the cells is not necessarily to block the effect of the cell intrinsic effect of PDL1. They might or they might not. So it's an open question. The different antibodies bind to different epitopes on PDL1, and so predictably their effect is variable. I hope I've answered the question.
Yeah. So the next question is from Jonathan Frieden. Sounds more of a philosophical question. It's pretty clear why cancer cells wouldn't do well in a hot tumor microenvironment, but can you say why again a cold environment is also disadvantageous?
Well, if you mean by cold, less expression of PDL1. So this is a guess. So it's an assumption on my part, I must confess. So I think by one mechanism or another, cancer cells have got to prevent themselves being killed by CD8 T cells. A major mechanism is to use the PDL1, PD1 interaction. But it's probably not the only mechanism. And I'm not an expert in this area, so others may well know more specifically how this might happen. So my guess is that if cancer cells don't express PDL1, they've got to find another way to resist being killed by CD8 cells. That's a hypothesis, but it seems a reasonable one.
Mm-hmm. Another question from the same person is the clinical trial issue with Roscovitin about funding or some other risk or business reason?
Well, I don't know about business reasons. It's not my business, it's somebody else's. But I think the bottom line is at the moment, the company seems unwilling to allow us to use the drug in a clinical trial. And I'm not quite sure why. We're continuing to talk to them and hope that they change their mind. All
right. So I guess this question was related to one of the schematics in your final slides. This question is from Chang Zhang. How does CDK5 regulate FBXO22? And you had a couple of hypotheses. Maybe you can elaborate on
Well,
it.
we don't really know. There are phosphorylation sites on CDK5. I'm sorry, on FBX. There are several phosphorylation sites. And so a couple of them lie in CDK5 consensus sequences, and so we're examining those first to see whether those are hopefully a direct target. I think that the simplest hypothesis and therefore the most likely one, is that CDK5 directly phosphorylates FBX, and that that phosphorylation, as well known, phosphorylations target proteins for E3 ligase dependent polyubiquitination. And so that's a likely hypothesis, but we don't yet know the site. And we haven't proven for sure that there's a direct interaction. There could be something in between.
All right.
That's as far as
Bill Forrester is also wondering the same thing, but adds on to ask, Roscovitin can inhibit many kinases, so wanted to ask about rescue alleles of CDK5 that would no longer be sensitive to inhibition.
Yeah, so, sorry, what was the last part of the question? Say again. What about CDK5? Yeah, go ahead.
So Roscovitin can inhibit many kinases. So wanted to ask about the rescue alleles of CDK5 that would no longer be sensitive to inhibition, but would still act to regulate FBX022.
Oh, okay. So I don't know about that. But CDK5 is the primary target of Roscovitin. It's absolutely true that it does inhibit other kinases, and that was the basis upon which it was used so far in the clinical trials, focusing on the activities against other authentic CDKs, if you like, rather than against CDK5. And that may be contributing to the anti-tumor activity even in our experiments. We absolutely don't know that. So, you know, using alternative CDK5 inhibitors would be a way to explore that. But you can see that at least in our cell culture experiments, CDK5 knockdown is very effective in modulating PDL1 expression.
All right.
So at least that works.
So next question, I think you may have answered this, but do you think anti-tumor effect induced by Rosco plus anti-PD1 is due to PDL1 downregulation induced by Rosco?
Yes, that's the hypothesis. It makes sense, you know, because yeah, it makes sense. Makes sense to me that if you're inhibiting both sides of the interaction, you're blocking PD1 and you're downregulating PDL1, it should make the effect greater.
Stephanie is wondering whether PDL1 mRNA levels are downregulated upon CDK5 inhibition, or is it just at the protein level?
Looks to be just at the protein level, yeah.
All right, Ed Frisch is asking, does Roscovitin or other inhibitors of CDK5 affect PDL1 expression in immune cells, especially dendritic cells?
A very good question. And it could be contributing to the in vivo anti-tumor effect. We're well aware, the work of several labs has shown that PDL1 is present on especially myeloid cells in the tumor microenvironment. And those cells could also be helping to inactivate, to block PD1 on the CD8 T cells. So it's a very strong possibility. We haven't looked at that yet, but it seems very likely to me.
Right. So this is an interesting question. Rami Reddy is asking, since luciferase has a T cell epitope, will those mice protected by the combination therapy are resistant to parental LLC cells?
We don't know the answer to that. So this is at a very early stage. The experiment I showed you was the first one that we did. We were pretty excited by that. We've now repeated it with very similar results, varying the conditions, you know, to see if we can increase efficacy and decrease the lethality of which you saw in response to Roscovitin alone, which we don't understand. But it's very early days, so we haven't looked at that.
All right. Nitya Balasundaram says it's an interesting talk. Thank you. Any comments on how the presence of interferon gamma in the tumor microenvironment enforces PDL1 expression only on the malignant cells, not on the immune cells? There are recent works showing PDL1 positive immune cells, such as NK cells, showing that PDL1 positive NK cells are highly cytotoxic when compared to non-PDL1 positive NK cells.
No, that's great. Yeah, sorry, go ahead.
No, that is the end of the question.
Yeah, so I don't know. That's fascinating information. So yeah, I think I made it clear in some of the data that I showed you that type 1 interferon beta, also can upregulate PDL1. But as you've mentioned, the major cytokine regulating PDL1 expression is interferon gamma. And how that works in different types of cells is, I think, a very interesting issue.
All right. Yong Teng is asking, what are the cell phenotypes of PDL1 inhibition?
PDL1 inhibition, that's reducing the expression. Is that what you mean? I assume so. So Roscoe-Routine or knockdown.
Yeah, I guess maybe the question is related to what are the cellular phenotypes that respond to PDL1 inhibition? Not sure.
Yeah, I'm not sure. I mean, first of all, we've only tried this, of course, on cells that express high levels of PDL1 so that we could, see the phenomenon more clearly. There haven't been any gross phenotypes in the cells, apart from the properties that I showed you, change in the response to interferon, change in sensitivity to DNA damage, those sorts of things. But the cells grow pretty normally.
Okay. So Hoxton is asking, low levels of interferon help tumor cells to survive, which could be mediated by some immunological mechanism mediated by CTL or macrophage-mediated phagocytosis. So how is this mediated?
Yeah,
is the question.
So I don't know. As I've mentioned, it's very obvious that interferon present in the tumor microenvironment can come from a large variety of sources, not necessarily from the tumor cells themselves. But interferon is kind of interesting in that way. There's an old literature showing that autocrine interferon produced in which the cells are responding to the interferon that they make is very, very difficult to interfere with, because even antibodies against interferon are not effective in blocking that autocrine response, probably because the interferon is coming out of the cell and the receptor is right there on the same cell, and it's tough to block that. So the biological effects of interferon produced by the cells themselves may be much stronger than the biological effect of exogenous interferon, which may or may not be present at high levels. So it remains to be seen whether in the real tumor microenvironment, what is the most effective source of the interferon that the cancer cells apparently need. But they can do it themselves.
Ines is wondering whether retro elements can be responsible for the IRDS expression in cancer cells.
Absolutely. And Andrei Gudkov has published a couple of wonderful papers, including one very recently, showing that activation of line elements in cancer can activate tremendous interferon synthesis by producing either RNA or double-stranded RNA or DNA or both, and that's a very important mechanism in cancer. A couple of his recent papers are very wonderful and worth reading on that topic.
Yeah. There's a question from Hannah Meredith. Has the baseline level and temporal dynamics of interferon production been characterized in different cancer cell lines? Just trying to get a sense of where the high versus low levels of interferon stimulation used in the experiments are relative to normal levels experienced in a cell.
Yeah. Yeah, I think it's likely that different cells will be producing different levels of interferon because at least the autocrine expression of interferon will be very dependent, of course, on whether the stimuli are there, either double-stranded RNA, which could come, for example, from line elements, or DNA, which can come from possibly a variety of sources, probably even present at a very low level in normal cells, but there are mechanisms in normal cells that block that response. So I can easily imagine different levels of DNA, intrinsic DNA damage in different types of cancer and different individual cancers, and therefore different levels of interferon production. So it's a good question, and we have very little information on it at the moment.
All right. We have a couple more questions, but we're over time, and I think if you can very well find Dr. Stark's email online, and you can shoot him these questions directly. Thank you, Dr. Stark,
Okay.
for taking the time to present wonderful work, and take care. Thanks.
Okay, thanks, Balaji. See you soon.
See ya.